Evening, guys. Let's get started. Um, I got a question, or rather a request, just now in chat about the phenol coefficient dilutions explanation. Um, you can I explained that last week, or I guess the week before we had the test. Gurley, you're asking. Like my last lecture 2 weeks ago before we wrote the midterm, the last 20 minutes or so, I was explaining this particular slide, which is basically how to read the dilutions of the phenol coefficient lab. So, in addition to whatever you're being explained in the lab, so you can refer to that lecture. You can go back to that lecture and look the last, like I said, about 20 minutes of the lecture, maybe even less, 15. You very briefly, like I said, it's the same thing. Like my explanation is going to be exactly the same. You look for the critical dilution for your test disinfectant. You identify your critical dilution for the phenol, and then you divide one by the other. You divide critical dilution of phenol by critical dilution of test disinfectant. That gives you the phenol coefficient. Uh, Critical dilution, how do you establish that one? It's the highest dilution, which kills at 10 minutes and doesn't kill at 5. Okay, so if you look at these two columns, there are two dilutions for this particular hypothetical case for the test disinfectant. There are two dilutions that satisfy this, or partially satisfy, I guess, this requirement. So two dilutions that kill at 10 minutes and don't kill at 5. It's 1 in 600 and 1 in 700 because, as you can tell from the table, for both of these dilutions, 600 and 700, 1 over. It kills this dilution, both of these dilutions kill bacteria at 10 minutes, so there's no growth, and G, no growth. At 1 in 700, but there is growth, positive growth at 5 minutes exposure. There's also no growth at 10 minutes for 1 in 600 dilution, and there is growth at 600 dilution. So which one of the two do we pick? Like I said earlier, the highest of the two, the highest dilution. The highest dilution, remember, is the one that gives you higher level of dilution, which is in this case 1 in 700. 1 in 700 is higher dilution than 1 in 600. So make a note, like if you were to solve this, 1 in 700 is your critical dilution for test disinfectant. And then for phenol, same deal. Highest dilution that kills at 10 kills all the bacteria at 10, doesn't kill at 5. So which ones kill at 10 and don't kill at 5? It's these three, right? 100, 105, and 110. In all three cases, bacteria is fully killed at 10 minutes, but still alive at 5 minutes. Kills at 10 minutes, still alive at 5 minutes. Kills at 10, uh, still alive at 5. Doesn't matter how much is alive. We're not quantifying anything. We're just looking at the turbidity of the test tube. If the test tube has at least some turbidity, any turbidity, that is considered to be a positive result, positive in terms of growth, right? If there's no turbidity whatsoever, a completely see-through clean test tube, only then you write it down as a negative result, no growth. That's pretty much it. So which of the three is the highest dilution? Obviously the 1 in 10, because it's a higher level of dilution than the other two. And then as I showed here at the top of the slide in the title, you divide critical dilution of phenol, which is 1 over 10, by 1 over 700. Basic math, the ratio flip, since you're having two ratios here, and you end up getting 700 over 110, and it gives you approximately 6.36 phenol coefficient. Okay. What does this phenol coefficient, what does this number tell you? It tells you how much more efficient your phenol is, your test disinfectant is with respect to phenol. So it's roughly sixfold more efficient than phenol in my particular example, obviously. That's the only information you get from the phenol coefficient. You don't know the percentage kill as we discussed it. Remember, I was explaining you this stuff before the test, like these calculations for the percentage kill, the log 10 reduction, the log killing. You don't have any of that information because that's not what the phenol coefficient is supposed to tell you. So you don't care for that. You just all information you the only information you collect, the only piece of information you collect is the how much more efficient your test disinfectant is with respect to phenol. That's it. It's a semi-quantitative method, so it's not the best. It's not the most, I guess, informative, but still viable for certain applications. Uh, in the lab, since you guys, I guess, did not perform week one, you were not setting up this kind of a table. You were immediately jumping into week two. So I don't know exactly how your lab instructors set it up, but you were there could have been some modifications and whatnot, but you are basically looking at a range of. I mean, for specifically for the labs, you have to refer to whatever you did in the lab. But just remember that you are not setting up this table for the lab. You were probably given already hypothetical values for phenol coefficient, and then you were setting up the second week, and based on those hypothetical values, you would get a range of results with growth or no growth in the test tubes, and you would have to identify which of these hypothetical values actually corresponds to a true value of the phenol coefficient. Why hypothetical? Because again, we didn't set up this table, this part of the experiment in week one, so we can't really calculate. We don't have the basis to calculate the phenol coefficient. You just assume a range of values, and then you set up your second week in use dilution with all of those. Again, I'm assuming that's because there could be some modifications to the setup, so that's usually how I run it. I can't vouch for every single lab section out there the way it happened in the lab last week. So for lab related questions, especially the ones that could have been modified, it's better to just talk to your lab instructors. But as far as how to read the table from the first week, or how to identify the phenol coefficient based on on these kind of tables, that's what we have here. Yeah, in this slide on this slide. Before we move on, and then again, Gurley, everybody else, this was explained as well, pretty much the same way I just did it just now, 2 weeks ago before the test. As far as the test is concerned, quick, I guess, announcement. I posted the grades. Obviously, everybody's seen them. Um, you still have an option of taking a look at your test, a much better average this time around. I think I posted it was like 60 something percent, 66 or 67%, which is much better than last time. I mean, always could be higher, but that's 66 usually is okay result for the second midterm as an average. Some people did much better, some people did worse. It really varies, obviously. But um, you still have a chance to take a look at your test, at your results. A bunch of people already emailed me, like, I don't know, 15-ish or so, um, expressing the desire to go over their test. I was a little bit sick this week, so I couldn't really schedule any meetings this week. I wasn't in college this week. So next week is when we're going to meet up with everybody who emailed me. I let them know. If you haven't emailed me yet, but you still want to take a look at your test, there's still time to do it because, again, nothing has been scheduled for this week. Everything will be happening next week, I guess, hopefully. I mean, I should be fine for next week. Maybe not on Monday, but like later in the week. So if you want to take a look at your results, go over your mistakes, email me. over the weekend, this coming weekend, today or the weekend. And when you email me right away so that we don't have to email back and forth too many times, indicate in your email all of the days of the week and all the time windows roughly that work for you to see the test. So, for example, email me with information that I'm available to look at my test on Tuesday from 12 p.m. till like 5 p.m. or like when as well as Wednesday like in the morning from 8 a.m. till like 11 a.m. like specific details. Right. And for every single day, don't just give me one day option because, again, it's not about me so much. It's about the fact that there's a lot of students and managing every single one is like when there's like 15 or 20 people, it's pretty hard, right? You know, like I don't want to be in college every single day of the week, so I try to pool everybody in more or less at more or less convenient times. So give me all your availability to see the test. And then once I have everybody by Sunday, I guess, once I have everybody's day availability, I will email you back telling you when you can show up to see your test in the office on the third floor. See this office on the third floor. So you still have that chance, right? Like I said, it's since nothing happened this week, next week is going to be the day. Is going to be the week to do that. Next week is week 11, so you know the last exam is on week 14, so um, still time. There's still time. Um, as far as the final exam, so will happen on week 14. Um, we'll cover only the last part of the course, whatever we cover starting today, right? And until the end of the course, which is only like week 10, 11, 12, and 13, 4 weeks. It's going to be the end of this lecture that we're going to finish today, and it's going to be two more lectures on antibiotics and antibiotics resistance. That's it. So 2.5 lectures towards the final exam, plus the calculations as always. Same format, same style, same everything. Calculation questions, about 10 of them, 2 points each, all that stuff. Okay, the final exam is going to be the heaviest in terms of the grades, as in terms of the marks. So the first midterm was 15%, the second midterm was 20%, the final exam is 25, right? So it adds up to 60. So 25% is almost half of all the marks allocated to lecture component of the course, 25 out of 60, right? Almost half. So that's a lot. So even if you didn't do so well in the first two tests, uh, there's still a lot of marks left. Uh, so do your best on the final exam. We can, again, if you have questions, you can always show up to the office next week. Again, send me an email. Don't just show up randomly. I'm normally not in college this semester at all. So if you just show up there, you're never going to see me there. Send me an email, and we're gonna set up the time, and then we can look at your grades and do the calculation to figure out how much you need to score in the final exam to pass the test. A few people emailed me by now um, with like lower people with lower grades expressing concerns and everything. In the middle of the semester, I can do anything. Uh, we have to see all your grades first, right? That's what all the evaluations are for. Uh, Once all your grades are in, including obviously the final exam, the labs as well. I mean, even though they don't overlap, you have to pass, as you know, individually this theoretical and lab component. But once all the grades are in, then once I have a complete picture, I take a look and see what can be done, what cannot be done. So for now, as always, in the middle of the semester or whatever, close to the end of the semester until the semester is over, the best you can do is just get as good grades as possible. Uh, Collect as many grades as possible from the remaining assessments. And once everything is in, like I said, once we have a complete picture, then we can decide something. Um, The best option, like I said, not an option, but the best strategy is just to get as many grades as possible. The closer you are to a passing grade or in general a better grade, I always try to help students, but I cannot like, you know, give you too many marks, so to say, right? It's not like I can just give students 10% or something out of the total grade just so that that person can pass or get a B plus instead of B or something along those lines. Like that's obviously too much. It's academia. You can just, if that was the case, then why would we have any assessments in the first place? Just give everybody an A plus or something, right? So yeah, that's the strategy. Uh, but again, if you want to take a look at the exam, which I strongly encourage all the time, not that many students show up, considering we have 240 students in the course or something along 234, I think, a little bit less than 240. Uh, Only a small fraction shows up, honestly, very small fraction. So it's up to you. Again, I'm not forcing anybody, just saying that it's always beneficial when you look at your mistakes. Trust me, speaking from experience, many years now, I've seen thousands of students, thousands, not just 1,000, but thousands. Uh, Sometimes people make mistakes because they didn't know the material specific. Sometimes students make mistakes because they misread the question, like a specific question or two or whatever being misread. Sometimes people make mistakes, quite often actually, because they tend to overthink the question. And that's like part of you know how we think. People sometimes put too much into the question that is being formulated, and like it's almost like a part of mentality, so to say. Um, so for cases like this, the reason I'm mentioning this is because this latter case where people overthink the question, like I mean, the previous two, when I said, I mean, if you're not prepared for the exam, the very first one, it's just there's nothing that can be done, right? So I'm not really stopping on that option. It's pretty straightforward. You got to study for it. Second one, when you misread the question, usually students don't misread too many questions. Maybe one or two that can happen, but usually not that many. The third option where people overthink the question is where usually students land a lot of mistakes because when people overthink the question, they tend to overthink a lot of questions, not just one or two, but it's like consistently happens. They read into it too much every time they see a question. So that kind of stuff, that kind of problem can be avoided if you, but for that, you have to show up and actually take a look at your test and try to answer yourself, first of all, why did I make that mistake? Did I misread it? Did I misunderstood it? Did I just not know the answer? Or did I like overthink it? And like I said, though, this overthinking can lend you a lot of mistakes because that is something that is like almost part of a personality. People tend to do that a lot every single time or every second time. So that is a lot of questions, can be a lot of questions. So for that reason, always use this opportunity, take a look at the test and go over every single question that you where you made a mistake and ask yourself like, why did I pick this answer here? I wouldn't have that answer for you. You're the one who are doing it, right? So, um, I mean, sometimes students show up and they just compare the answer sheet that I give them with their own answers. They are basically this way they're verifying whether the machine made a mistake or not, which can be done. I guess there's nothing wrong with that. Very rarely so, but sometimes machine can make like a mistake, a single mistake on a Scantron out of like one Scantron out of 200 or something. That can very rarely happen. But that is not what you should be doing when you take a look at your test. You should not be verifying the machine. You should be answering that question to yourself. Why am I making these mistakes? What is the reason so I can avoid that next time? If it's just because you didn't know the answer, then I mean, you just got to study better. It's as simple as that, as obvious, or maybe not so simple, but that's just an obvious answer. But if it's something like overthinking, you need to address that. So yeah, Send me an email. Like I said, you still have a weekend. Next week is when I'm going to meet up with everybody who already emailed me. So get into that line, so to say. There's still room. There's still time. Alrighty. Back to the lecture. So, like I said, it's not going to be too long today. We're a little bit ahead of the schedule, so to say. We have what 3 weeks remaining, 10, 11, 12, 13. We have 2.5 lectures to cover, so we should be fine. So this phenol coefficient test that we looked at last time, one that you performed in the lab, this title here, the Schick-Martin test, is basically a slight variation of the uh, phenol coefficient test, um, which is common with tests in general with all sorts of setups in the lab. There's always going to be variations to one procedure. There's always going to be variations to any procedure. Um, the point I'm talking about this variation of the phenol coefficient test is to draw your attention to some of the methodological aspects of running any experiments, not just the phenol coefficient. So let's take a look first at what's the difference between the Schick-Martin test and the Rydell-Walker method. The Rydell-Walker method is the phenol coefficient, another name for this phenol coefficient method, the basic one that we looked at last time. The Schick-Martin one is the modification. It's run in exactly the same manner, except for, as this table highlights some of the differences, the media volume can be adjusted from 5 to 10, for example. Instead of using distilled water as a diluent for your test disinfectant, you can use water that is contaminated with either yeast or dried human feces. And it's a very common thing to do. People use, in the experiments, they used yeast matter or dried human feces as um, I guess, the emulator to emulate, to mimic contamination of your dilution with organic matter. Okay. So instead of using distilled water, which is a perfect scenario, right? You apply disinfectant or you dilute disinfectant for your method with something, with a diluent that is already contaminated with some complex organics. And human feces serves as that compound, not the most pleasant one, but that complex organics that it emulates contamination, that mimics the contamination of your solution with complex organics. Sometimes people also use albumin, solution of albumin protein, to also mimic the contamination. Contamination not by bacteria, don't get me wrong here, but contamination by organic matter. Why do people do that? Why do scientists do that? We discussed this before, because efficiency of disinfectants and efficiency of the disinfection step um, may depend on presence of organic matter as well as inorganic matter, all sorts of organic, inorganic contaminants that can be present on the surface, right? And that's why last or 2 weeks ago when we discussed this, I told you that the very first step of any disinfection procedure should be cleaning of the surface. So remember that CDS strategy: cleaning, decontamination, sanitation, CDS. So cleaning is always essential, or not maybe always, but often is essential. Why? Because if you don't clean the surface before you apply the disinfectant, if your surface is contaminated with organic matter or inorganic matter, it could be both, could be either, could be both. That contamination may severely reduce the efficiency of your disinfectant, right? So the efficiency of disinfection will drop, You will not kill, you will not eliminate all the bioburden you were supposed to eliminate. So for that reason, um, you want to clean the surface first with just soap, without any disinfectants or anything, just removing the dirt, as they call it, the dirt, which is usually a complex mixture of organics and inorganics. So that's why scientists use either a solution of the albumin, it says actually here, 3% dried human feces or yeast matter to introduce that contamination factor. And as we will study later today, as we go through a bunch of classes of disinfectants, not every disinfectant is equally efficient in the solution that is contaminated with organic matter. There are some disinfectants, some classes of chemicals that are disinfectants that are extremely efficient. They don't care for any contamination. They will kill bacteria, will they remove bacteria even in the heavily contaminated environment. So those are really nasty and really good disinfectants. They're usually very harsh disinfectants, but nevertheless, certain other disinfectants, for example, um, alcohols, the alcohol that we use to disinfect the bench. We actually don't use alcohol to disinfect the bench anymore. We use Dettol and citrus, but in the labs, it's very often where people use isopropanol to disinfect the bench or ethanol to disinfect the bench. So for the alcohols, um, we'll talk about it later today, but just to illustrate the concept, alcohols are not so good at disinfecting the surface that is actually dirty. So for the alcohols, you want to clean the surface first and then apply that. So that's the reason people use these conditions for the phenol coefficient, modified phenol coefficient tests. As you can see, the incubation temperature can change. Okay. The standard method was using 17.5, 18 degrees Celsius, 20 degrees Celsius temperature. In the Schick-Martin test, they bring it up to 30. Another factor that can be different is the organism. Salmonella typhi is usually the go-to organism for the Rydell-Walker method. The Schick-Martin test has been standardized for Salmonella typhi as well as Staph aureus, so an additional organism. Sampling times, we looked at, or theoretically we looked at, we didn't actually do it, but we theoretically assumed 5-minute exposure, 10-minute exposure for week 1, as per this table that I was explaining earlier. But there's also other time periods that people are considering. For example, Schick-Martin test specifically standardizes the experiment for 30-minute exposure. That actually changes a little bit the formula, how the critical dilution is being calculated, but don't worry about that. You don't need to know that. And then as you can see here, as a result, the calculation of coefficient can be calculation of the coefficient can use different formula, not necessarily 5 and 10. But as you can see here, it could be killing at 7.5 minutes divided by phenol dilution. So all sorts of approaches. Don't need to know those. Don't need to memorize the table. What you need to understand, where I'm leading this to, I mentioned a couple of times as I was going over this, the word standardization. The Schick-Martin test is standardized at 30 degrees. Schick-Martin test is standardized for Staph aureus. So what does that mean? Whenever you set up any sort of experiment, phenol coefficient is just using it as an example to illustrate the concept. When you set up an experiment, you trying to answer some sort of scientific question, right? In this particular case, the scientific question was, what is the phenol coefficient? How much more efficient my test disinfectant is with respect to phenol? When you run the experiment, you get certain values, right? Phenol coefficient of 6, whatever, 5, 10, whatever it happens to be, right? Once you're done with the experiment, the first question the scientist, or a good scientist anyway, should be asking himself is this value that I got trustworthy? Is it actually a trustworthy value? What if it's a random value? Somebody can come up to you and say, well, how do you know this is actually trustworthy? Maybe it's just a fluke result, a random result, right? So what do you do next to avoid these accusations, so to say? You should know by now, you run your experiment in multiple replicates, right? You should be familiar at least with the basics of technical replicates, biological replicates. So if not, actually, I guess I can mention it here briefly. So whenever you say do, uh, okay, let's go back actually to phenol coefficient, good example. So in the lab last week, or whenever you ran this 2 weeks ago, you were setting up your phenol coefficient in 10 test tubes, right? You were putting the penicillin into 10 test tubes, exposing it to disinfectant, a certain dilution, and then uh, moving the penicillin into the recovery media. So those 10 test tubes that you were using are the 10 technical replicates, because all 10 test tubes that you set up were set up exactly the same way. There's nothing uh, different about them, right? It's the same dilution of uh, disinfectant. Different groups were getting probably different dilutions, but within the same group, you were running the same dilution in all of your 10 test tubes. You were exposing it to the same time, about 10 minutes should have been, and then you were moving that penicillin into the same media, right? Recovery media. It had to be either lethine broth or nutrient broth, depending on what kind of disinfectant you were using. So those 10 test tubes is basically 10 copies of the same result, right? And those kind of copies we call technical replicates. Another example, you are doing the spread plates, for example, setup. Uh, it's never a good idea to plate from your test tube after all the dilutions, whatever you had to do. Um, it's never a good idea to plate from the test tube only once on a single plate. Quite a few students are still doing this or used to doing this in the project course. I don't know, probably still the case. I wouldn't expect this to completely get eradicated, but one plate is never good. That one plate is a one technical replicate. Uh, you always want to have at least three, better five. If we're talking like serious experiments, serious setups, people are usually looking for five replicates of the same result. So why does it have to be three, five, or even 10? Because again, one result doesn't matter how good it is, doesn't matter how expected it is. It's also a very common misconception where students setting something up, and obviously quite often they do have an idea what they're supposed to get back, like what kind of result they're supposed to get. When you're doing the spread plates, you aim, for example, for 100 colonies, and then so I should be getting about 100 colonies. And if you end up getting like 90 colonies, you're super happy, but one plate, one result is always bad, because that one result can always be a fluke result, as they call it, right? A random result, your lucky result. Or it could be the other way around. It could be an unlucky result. You expect to get 100 colonies, you do one plate, and you got, let's say, 5 colonies. And then you start questioning, okay, what is wrong with my technique? Where did I make a mistake, and so on. But that one plate could have been just a fluke. If you plated actually 3 or 5 or 10, you could have gotten 9 plates with like 90 colonies or whatever, 80 colonies, 90 colonies, 110 colonies, so around 100. And only one plate, for whatever reason, we're not going to discuss the reasons right now, but for whatever reason, one plate was a fluke. So for that reason, reporting your results based on just one technical replicate is always a bad idea. And anybody half knowledgeable in science will never appreciate this, because those kind of results are not trustworthy. That's where the whole concept of stats and running the statistics behind your results come into play, right? Those error bars and stuff. The analogy I usually give to students to get a better grasp of this idea imagine that we're sitting in the lab and then there's a student coming in to the lab late, and the student says, I just saw a dinosaur on the street, right? Would you trust that student? Probably not, right? Because he could be joking, whatever. So that's what we are one technical replicate. Then imagine, um, so to give you a proper, I guess, analogy, so the same student, very important here, says, I saw the dinosaur. So you hear that, you don't trust that, you ask him again. You saw the dinosaur in the street, so you ask him again. He says the second time, yes, I saw the dinosaur in the street, right on the corner, whatever, the bus stop, there was a dinosaur. So that second reply is your second technical replicate, right? And then again, you go, I don't believe you. Repeat it again. Did you see a dinosaur like one of those from the ancient times? It's like, yes, I did see that kind of a dinosaur in the street. So that would be the third technical replicate. Uh, so when there's one technical replicate, it's not really trustworthy. When there's three technical replicates or five, or you ask that student the same question 10 times, it's technically more trustworthy, right? Because the student confirms the same answer multiple times. So it's not he heard the question right. He didn't confuse the whatever the words and or something. But as you can guess from this example, it's still not very trustworthy, right? Or could be not quite trustworthy. So using this analogy, what would you usually want to have in addition to that single report? You want to have not the same student, report a dinosaur in the street 5 times. You want a different student to come in and say the same thing, right? If a second student walked in, a completely different individual, and said, "Yes, I saw the dinosaur also on the street, right, by the college bus stop." Now, that second report from a second individual would be, in my analogy, an example of a biological replicate. When you basically get the same piece of data from a completely um independent source. Obviously, in my analogy, you can always say, well, they're basically joking together, whatever, you know, obviously we're skipping that part, but it's just an analogy. But uh, a second individual independent source is the analogy here for the biological replicate. So what is a biological replicate uh, in in the lab, in the actual lab settings? So if you're running your experiment, let's say your um, phenol coefficient, the way you did it 2 weeks ago, and you were provided a phenol coefficient, a hypothetical value of, say, phenol coefficient of 6. You set up the experiment once, the way you did it. You set up 10 test tubes with the same dilution of your phenol. Those 10 test tubes are your 10 technical replicates. You get the result, you record the values, you do all the math. Then what you do next is you scrape everything. You change the media, you change the diluent, you change the preparation of your test disinfectant, you change the glassware, everything. And then you set up the same experiment with the same phenol coefficient value that was given to you from complete scratch. Okay? With another 10 technical replicates, with another 10 test tubes as technical replicates, but completely new reagents, freshly prepared, new media for recovery, nutrient broth or lectin broth, freshly prepared, very important. All fresh, all new, the same values because you're still testing the same value, right? But all the reagents new. So that second setup would be your second biological replicate. Okay. So when run properly, uh, any experiment, not just the phenol coefficient, I'm using that phenol coefficient as an example. Uh, anything really, anything that you want to be actually trustworthy, any result that you want to be trustworthy, you have to run it in multiple biological replicates, at least 3, better 5, and multiple technical replicates, at least 5. Only then, and after you do all of that, right? Very important. Uh, you're expecting to get the same result every time you set it up, or almost the same result. There might be some fluctuations, obviously, but again, those fluctuations, after you analyze them statistically, should not be any significantly statistically different. Obviously, things will always fluctuate. It's in the physical world, you're never going to get exactly the same value unless you get lucky. So there will be fluctuation, but it should be statistically insignificant, those fluctuations. So only in that case, when you've set up multiple biological replicates and each biological replicate had multiple technical replicates, and every single time you do that, you get the same or almost the same value, statistically insignificant deviation from the same value. Only then you can claim that you standardize the procedure, and which means in its case that this procedure, the way you set it up, the way you do it, the way you run it, is actually trustworthy to answer your scientific question. In our example, if obviously you didn't do it this detailed in the lab, but if you've set up multiple biological replicates, multiple technical replicates, several times, and then you get every time you run it, you get a phenol coefficient of say 6.36 or around that value, only then you can say that the way I'm setting up this experiment with these dilutions, with this organism, with these temperatures, right? Referring now back to the table here. With this particular organism, in our case, it was Staph aureus. I believe you used Staph aureus in the lab. At that particular sampling time, 5 minutes or 10 minutes, whatever was it, at that particular temperature, was it 20 degrees Celsius or something? Now, this experiment now is standardized for these conditions, which means that you can use this experiment to answer that particular scientific question. And the results that you get from that experiment will be trustworthy according to your data, according to your setups. You're not going to get a random. So you basically, by standardizing, you're guaranteeing, right, the reader that you're not going to be getting random values every time you set up your experiment. Because if you're getting a phenol coefficient of 6.3 one time, and then you set up the second biological replicate, you get a phenol coefficient of 20 for the same setup. You set up the third time, you get a phenol coefficient of 1. That is not trustworthy data. So anybody who looks at that data will say, why should I use your experiment or your methodology rather to answer this question of what is the test disinfectant phenol coefficient if your methodology doesn't give me consistent data? It's random every time, right? It's obviously not trustworthy. You have issues with your setup. So you don't want that. So when people create different variations of the test, referring back to this table, Rydell-Walker method, the Schick-Martin method, they, for different reasons, for their different applications, they sometimes have to change different conditions within their methodology. Just like in the Schick-Martin test, they want to add different organisms, not just Salmonella, the one that was used originally, but something else, Staph aureus, E. coli, Pseudomonas, Acinetobacter baumannii, or whatever have you. People, for whatever reasons, we're not going to discuss those, decide to change the temperature of the method, right? Because for their application, 17.5 or whatever, 20 degrees standard room temperature doesn't make sense. They need 30 degrees. So the point is, every time you change any of these parameters, you have to show that the method, the viability of the method, the reliability, the accuracy of the method still holds. The method is still giving you trustworthy data. So whenever you change these parameters, you have to standardize the method. You run it multiple times, as in several biological replicates, several technical replicates, and you look at the data you get. If the data is consistent, if the data is stable, so to say, if the data gives you, for the same conditions, more or less the same value, then you can say, okay, for my changed conditions, this method or this methodology is still applicable. It gives us trustworthy data. What can happen and what very often happens with experiments, somebody, to stick to this table, somebody gives you Rydell-Walker method and it works. But then for whatever reason, you decide to run exactly the same method, but with Staph aureus instead. If you just blindly run it with Staph aureus without standardizing it, you may run it perfectly fine, but the data will not be consistent. You will run it on the same phenol coefficient. You will run it under the same conditions. But you run it once, you get a phenol coefficient of 6. You run it second time, you get a phenol coefficient of 20. You run the third time, you get a phenol coefficient of 1. So that would mean that if you just changing the organism itself may have that impact on the methodology. Okay. Because a different organism is a different organism. It has different structure. It has different resistance. It has different, whatever other physiological parameters. It may not like the media that you're using, or it may not be susceptible to that particular disinfectant. So something that worked for Salmonella in our example may not necessarily work for Staph aureus. It doesn't necessarily also mean that it will never work for Staph aureus. It may, in some cases, require small tweaking to methodology. For example, you will have to change a disinfectant because a certain disinfectant that was used for Salmonella may just not have enough power, so to say, against Staph aureus. So you change the disinfectant and you get now meaningful data. So this logic that I just kind of over-explained applies to every single factor, or almost every single factor. Temperature, contamination with organic matter, yeast, human feces, and so on. Even these contaminants, we talked about 3. Albumin, 3% dried human feces as, mimicking the organic contamination, or yeast matter. When you introduce each one of the 3, you might want to standardize the method because albumin, for example, solution of albumin, may have a completely different impact on efficiency of your disinfectant compared to that impact of dried human feces, because dried human feces or yeast matter, yeast lysate, is made of different chemicals, right? It's made of much more proteins than just albumin. So it may interact differently with your disinfectant. And as it interacts differently, it may, affect your tests, your methodology, and the data that you get from that methodology. So every time you change these parameters, any of the parameters in the experiment, do not assume that the experiment will be still, the methodology will still be valid. It may remain valid. It may actually require some tweaking, some standardization to use proper scientific methodology, terminology. Okay. Very common misconception, very common source of mistakes for students, and not just students. I've seen PhD students doing the same mistakes. It's just overall, I guess, proper understanding of science. Standardization is not, something you can neglect. Every data that you get, every piece of data you get has to be reliable. It doesn't just become reliable automatically. It has to be made reliable. Any experiment, phenol coefficient is no exception, is basically a system where you have multiple different parameters, multiple different moving parts, so to say, to it. You change the temperature, you change the organisms. The organisms can be so different. One thing you're working with E. coli, the other thing you're working with Pseudomonas. They have different physiology. They have significant differences in the structure of the cell wall and membrane and whatnot. There could be different plasmids present in those organisms. Those plasmids can confer resistance, different levels of resistance to different types of disinfectants, right? So this is just to name a very few. There could be a bunch of variations. So every time you run something and you change and it works for that particular setup, if you change anything in that setup, you have to first prove that methodology still holds, okay, for your new parameter introduced. And that's why people, change methodology, they change the organism, and they change the methodology accordingly. So this is more of a general, I guess, concept in science, but can be illustrated with these two uh, modifications of the same basically procedure. Rydell-Walker method as well as Schick-Martin test. Adding complex organics may require having some sort of a I don't know, a detergent may be added to the solution so that your results remain valid. So all sorts of things. Don't just automatically assume a very, very, very common misconception by lab technicians and people neglect they underestimate the impact that the differences in these parameters, what kind of impact they can make on your data. Alrighty, so, or for example, you change the temperature again to illustrate this better. If you change the temperature from 20 to 30 or from 20 to 40, you think, well, what's going to happen? I just elevated the temperature a little bit. But what if at that temperature, at 40 degrees, your disinfectant, that particular disinfectant you're using becomes unstable, or it becomes much less stable than it was at 20, and then completely everything gets impacted, right? So your data may completely get unreliable. Alrighty, so hopefully you get the idea. As far as memorizing anything for the test, you don't need to memorize anything, any values specifically. I'm not going to be asking you, what temperature is being used for the Schick-Martin test. These things get changed a lot. You need to understand the general concept behind this, but not necessarily the details, like specific details. Those specific details are changing very often. Once you get to work with this kind of stuff at the workplace, then you will have to memorize the details. So this American Association of Official Analytical Chemists. So this procedure, the carrier test, was the test that you ran in your phenol coefficient lab 2 weeks ago. When you used the carrier, which was a penicillin, the penicillin was being exposed to culture, then you were putting it into the dilution of your disinfectant and all that stuff. This procedure, in-use dilution or carrier test, is basically aiming to answer one question: how a carrier contaminated with culture affects the efficiency of disinfectant. So the way we run it, it's a little bit simplified version of it. Obviously, we don't have time for it, but technically, you should be exposing your carrier, your penicillin, to culture for much longer, so that the culture has time to attach. The point here is the culture may attach to the carrier. And in some cases, people expose the culture to carrier even longer for a day or so, even longer, to allow the culture to potentially form a biofilm on the surface of the carrier. And then that carrier penicillin is getting exposed to disinfectant, and you look at the efficiency of the disinfectant. So again, those are all different modifications of this procedure. We looked at some most basic aspect. Depending on another thing is depending on when you use this carrier, depending on the material that the carrier is made of, that can also impact the formation of the biofilm. So one thing you're using plastic, the other thing is using stainless steel. A third option, I don't know if you're using some sort of I don't know, cellulose packaging material is something like that, right? So a cellulose-based packaging material. So all those factors may differ, and then they all require standardization, and they all have to be tested separately. As far as this is concerned, don't worry about that. We don't need these details. I'm not going to be asking you anything. Again, it's just showing you different, I guess, modifications to the procedure, whether you're using organic matter or you're not using the organic matter, you're using Staph aureus or using Salmonella typhi or you're using Pseudomonas. So just different modifications of this carrier test method. Um, sometimes, you're using different disinfectants. You have to adjust the media, the recovery media that you used. For those of you who are using citrus in the lab 2 weeks ago, you were using lethin broth, uh, to get rid of, to allow the cells to recover because lethin broth inactivates quacks, quaternary ammonium compounds. We will look at them later today. And citrus is based on quacks. It's a quack-based disinfectant. Those of you who are using Dettol, you're just using nutrient broth. So depending on the nature of the disinfectant, you're using different media. So again, when you use that media in the in the workplace, don't just automatically assume that one media will fit them all, so to say, right? Depending on the nature of disinfectant, you might adjust the media. So you need to know those things. At least you need to remember those things so you can, in time, ask yourself proper questions. Am I using the right media? Is this media, we were using nutrient broth, but is this nutrient broth good enough for something else, or the other way around? Is lethin broth good enough for quacks, or is nutrient broth needed for quacks? So you have to keep those questions in mind when you work in the real settings, and not just use the same methodology for everything. You change the experiment multiple times and you still use the same methodology, the same media, the same everything. That is a recipe for disaster, usually. Disaster or just unreliable results, or the results that will not make any sense. Alrighty, so that's that. So what are we going to do for the rest of this lecture? Let's make a break, I guess. Quick break, like 5 minutes. Um, we will study four, five, maybe five, six classes of chemicals, specific ones like alcohols, aldehydes, peroxygens, uh, as individual classes, separate classes. So for disinfectants, and there's going to be a lot of, not a lot, but again, some data to memorize. You will need to memorize all of these technical aspects for the final exam. So we will look at like 5, 6 classes, and that's pretty much going to wrap up the lecture. Okay. So we'll look at their mode of action, what they do, how they work, how they kill bacteria, how they lose and how they keep their efficiency, and so on and so forth. So let's make a break, I guess, um, and then we will come back to to the lecture. Vasima, to answer your question before we go, is PC a phenol coefficient only for phenolic-based disinfectants? No. It compares everything to phenol because phenol was traditionally used as a disinfectant of choice. But phenol coefficient is phenol in the name indicates that you're comparing everything to phenol. But what you're comparing to phenol can be anything. Can be citrus, can be Dettol, can be alcohol, can be whatever. Okay. Alrighty. Let's make a 7 minutes break and come back at 8:34. Let's make it 8:34. All right, let's get back to the lecture. So the first class of disinfectants we will look at are the alcohols very commonly employed. Um, Isopropanol, ethanol, some of the most commonly employed, two of the most commonly employed disinfectants. Um, so what do you need to remember? Like I said earlier, pretty much everything that is on the slides for the rest of the lecture, all these technicalities, they're kind of quite essential. So with isopropanol and ethanol, two of the most widely employed ones, you need to remember that isopropanol is more efficient than ethanol, is a better disinfectant than isopropanol or than ethanol. Uh, both disinfectants are active against vegetative forms, so including some of the more resistant species. Don't forget, I'm using Mycobacterium tuberculosis as an example of more resistant bacterial species out there. Not so it doesn't include just Mycobacterium tuberculosis, but in general, bacterial species that are famous for its high levels or elevated levels of resistance. So it's active against vegetative forms, but not so much against the spores, right? So they're not sporicidal, and that's their obvious downside. Um, Cytal activity drops drastically below 50% concentration. So what does that mean? That means if your concentration of your alcohol is below 50%, uh, you start losing the efficiency of disinfectant significantly. So you always want to keep it above 50%. Uh, However, jumping a couple of points here ahead, uh, alcohols illustrate well this one concept that is also quite commonly, there's a lot of misconceptions about it. Um, so as you increase the concentration of alcohol, the level of the cytal activity of disinfectant does go up. But uh, as you go in your concentration of alcohols, specifically alcohols above 90%, their cytal activity starts actually dropping again. So common misconception, people think, well, the higher the concentration, the more efficient is my disinfectant. Quite often it is. In case with alcohols, it is actually not. Above 90%, the uh, cytal activity starts dropping. Why is that the case? There's no magic here. Everything has its own mechanism in science. So uh, they actually figured it out sometime. They didn't know the answer right away, not so long ago when this was figured out. But water is actually essential. The presence of water is actually essential for the cytal activity of alcohols. So without water, certain concentration, minimal concentration of water in your alcohol dilutions, your alcohol by itself is not going to have any cytal activity. So 100% alcohol or close to that value is going to have very, very poor cytal activity. The water is essential for the formation, speaking of the mechanism, presence of water is essential for the formation of free radicals uh, from free hydroxyl radicals in the alcohol solution. And those free radicals, hydroxyl uh, radicals are the molecules, are the chemical entities that go and oxidize bacterial walls and membranes. So that is the working entity in the disinfectant. Free hydroxyl radical, OH, with unpaired electron. Very reactive, as you should know from chemistry, very reactive species. They have unpaired electron, and they are therefore very strong oxidizers. So water is essential for the formation of those radicals. Without those radicals, alcohol by itself has no magic. Okay? Keep that in mind. So usually people routinely prepare 70% alcohols for disinfection needs. So somewhere in between 60 and 90. But it's never above 90. Don't fall into that misconception. Um, One another downside of alcohols, we kind of talked generally about this earlier. Alcohols happen to have poor penetration into organic matter. So What does that mean? That means you must clean the surface before you apply alcohols. Otherwise, your disinfection efficiency is going to significantly drop with alcohols. They are getting absorbed by organic matter, and they cannot penetrate into organic matter properly. And therefore, bacteria will be exposed to much lower concentration of alcohol than it should be. And obviously, the efficiency will drop. Okay, so cleaning is essential. When you use alcohol, cleaning of the surface is essential. Like I said, isopropanol is more effective, roughly 2 times as cidal to bacteria, but it's also about twice as toxic to people. Keep that in mind. So good alternative to ethanol. Both have actually fairly limited virucidal activity, so they're not really good at killing viruses. Isopropanol has even lower virucidal activity than ethanol. Um, Once you get to other representatives of alcohols, like highly substituted aliphatic alcohols with uh, more carbon atoms in the chain, they lose their cidal activity. They are just commonly employed as preservatives. So they're much weaker antimicrobials than isopropanol and ethanol. Okay. The next class, commonly employed in pharmaceutical settings, a lot more efficient, a lot more cidal, are the aldehydes. Formaldehyde, so you know the general formula of the aldehydes, right? It's the presence of this group, CO, double bonded, right? And H, carbonyl group. So these are the aldehydes. Formaldehyde is the first representative, has only one carbon atom and hydrogen in place of the carbon chain. Formaldehyde is the simplest aldehyde, used to be employed a lot in pharmaceutical settings and in general as a disinfectant, especially in so-called fogging procedures. We discussed those previously in the course. Fogging procedure is when you completely seal off the clean room. Not every clean room has the capacity for it, but those that do, you completely seal it off, and then you just spray gaseous form of disinfectants, in this case, in particular the formaldehyde, into the room. Obviously, a very efficient procedure because gas is gas. It can reach any surface, any crevice, anything really, right? Much better than liquid. So very efficient procedure, but pretty expensive. Um, Quality control is essential. You don't want to be, you know, letting your personnel back into the room where you still have residual amount of, whatever gas, aldehyde, or whatever have you, still present in the room. So it's pretty expensive procedure. Not every facility has it, but it is a very efficient one. Formaldehyde is being phased out these days. It's not being employed often because it is considered to be carcinogenic when inhaled. So aqueous solutions are being employed more commonly. Formalin is an aqueous solution, 34 to 38% of aldehyde. You must have encountered this previously, must have heard about this. So know what formalin is. It's an aqueous solution of aldehyde, of specifically formaldehyde. Another shortcoming, I guess, of formaldehyde, the major shortcoming of formaldehyde, um, its activity is very limited in terms of temperature and humidity. So it loses its activity below 20 degrees Celsius and at low humidity, under 70% humidity. So you have to have very specific physical conditions in the room, in the clean room, for the formaldehyde to work. It can also polymerize. I think I have it somewhere on the slide. Yes, it can polymerize on the surface, so make a polymer made of multiple formaldehyde molecules, right? And as it polymerizes, it loses its activity as a disinfectant. So obviously that is not a good property. You don't want your disinfectant to lose the activity. Okay, so it's still being randomly employed here and there, but not a very common one. This guy here, glutaraldehyde, is a lot more common and a lot more efficient disinfectant. It's also an aldehyde, just a different representative. As you can see, has more carbon atoms. in the chain has 2 carbonyl groups. By the way, speaking of the formula, you do not need to memorize the formula. I'm not going to be asking you to draw or recognize the formula of glutaraldehyde. I'm just posting these for you to see, so it's a little bit more visual. But as far as memorizing, you don't need to. So glutaraldehyde is actually very common. It's a very common disinfectant. The mechanism of action, it is cell wall and DNA damage. So it kills the cell wall, oxidizes the cell wall, as well as does the DNA damage to the cells. So very efficient. And it's also very efficient against the spores. So it is sporicidal. So it's used as a chemosterilant, the chemical that is potent enough to sterilize the surface completely. Hence the name chemosterilant, right? So it kills the vegetative forms as well as spores, unlike alcohols. It kills Mycobacterium tuberculosis, so all the problematic species, all the high resistance, high level or elevated levels of resistant bacterial species. Very effective, doesn't require much time. 1 minute exposure kills vegetative bacteria, all of them. Spores obviously always require longer because they're a lot more tough structures, so that should be logical. But still, it is sporicidal. Alcohols are not, regardless of the time, unless it's some sort of very susceptible spores, but usually even against those, alcohols don't work. Glutaraldehyde is much better against the spores. Also, its efficiency, as you can see, is not significantly affected by organic dirt. So that means cleaning is not as essential. You can apply it to the surface right away. Again, in some cases, it's very convenient, saves time, saves money. And the only thing you have to remember is, and we discussed this before 2 weeks ago when we talked about pH impacting the activity of disinfectants, it has different profile of activity at different pH values. So if you want to make it active, you must use basic pH. So most active at pH of 8, which is basic pH. And as it says here, the stability of the glutaraldehyde is the highest at pH of 4. So it's acidic pH. So what am I referring to here is a very common situation with a lot of chemicals, a lot of biologically active chemicals in the lab. Their activity and their stability are on the opposite spectrum of pH. It's a very common case this year when the activity and stability are so far apart. So what does that mean? What are the suggestions? It's a pretty straightforward suggestion. So whenever you work with this chemical, you're obviously going to have a stock of it somewhere, right? So when you keep that stock, you want to have it stable. You want to have that stock kept at stable conditions, so it doesn't decompose. So the stock is being kept at pH of 4, but right before you use it, right, you make a solution of it and you adjust the pH to 8. A very important consideration. Something also to keep in mind about all sorts of chemicals you're using out there in the lab. They might be stored and they might be used at different pH values for this particular reason. So you keep it at 4, pH of 4, but right before you are ready to employ it, you must adjust the pH because if you use it at pH of 4, its activity is going to be low, and you're never going to know what happened, right? You know, in BI-209 lectures, they told me it's the best disinfectant, or it's not the best, but a good disinfectant, but it doesn't work for me. pH can be absolutely a factor. So the solution right before use has to be adjusted to 8. It's not as stable anymore, but you don't want it to be too stable at this point. You just want it to act quickly, kill bacteria, and after that, it's over, right? So you have to adjust the pH. Halogens, another common disinfectants out there, usually chlorine and iodine containing compounds. Chlorine is more effective. Iodine not so much. It's less effective. The most common representative you need to know for the test are the hypochlorites. These guys here, hypochlorites. Hypochlorites are the salts of hypochloric acid, HOCl. That's a hypochloric acid. Sodium and potassium salts of that acid are called hypochlorites. And the hypochlorites, easy way to remember it, it's the active compound, it's the active agent of your household bleach. That's what's in there. It's extremely potent oxidizer, HOCl, as well as the salts of that acid there. It's a weak acid, but it's a strong oxidizer. It's not the same thing. And because it's a strong oxidizer, it is obviously very efficient against the cell walls of bacteria. Most of disinfectants that we're looking at, you can mark it down on the side, would not be a mistake. Most disinfectants we're going to be looking at exert their effect. So they kill bacteria by oxidizing the cell wall and the essential proteins in that cell wall. And they disrupt the cell wall, they physically disrupt bacteria because of their oxidizing properties. Okay, so potent oxidizing agents, cell membrane damage, protein damage, cell lysis ultimately. Sporicidal at higher concentrations, so they are dependent on concentration a little bit, but they can be sporicidal. Undissociated HOCl form is more active than dissociated form. So this anion OCl, right, is anion of HOCl. So this is just me going back to that pH discussion we had 2 weeks ago. So if undissociated form is more active, what kind of pH you must use? You must use, considering it's a weak acid, you have to shift the balance from dissociated form back to undissociated form, right? So what kind of pH are you using? Low pH, right? Acidic pH. And it's also a very common case, undissociated form being more active than dissociated form, because free radicals that are actual oxidizing agents, the products of HOCl, the free radicals are formed much more eagerly from undissociated form of the acid. Once it's dissociated into the anion and cation, it's a pretty stable form. Forming a radical from this form is challenging. So HOCl, undissociated form, is the form to go. So that's why, for that reason, you have to use it at low pH, lowered pH. Some of the disadvantages, as it is the case quite often for more potent disinfectants out there, they're corrosive to the surfaces, they're corrosive to your skin, because again, they're oxidizers. They don't care, they just oxidize everything they can, so to say. Can be inactivated by organic dirt. Again, they're inactivated because they just oxidize everything. And oxidizing dirt in our applications is kind of defeats the whole purpose of killing bacteria, right? Instead of killing bacteria, you use your bleach or your hypochlorites in general to inactivate the dirt. So, which is okay, I guess, but the bacteria gets exposed to much lower concentration of the hypochlorite as a result, and they're fairly unstable. Halogen-containing compounds are fairly unstable, so they can decompose. Direct sunlight, temperature is a factor, so you always want to look into those details if you happen to work with these. Keep those considerations in mind. In general, like I said, this applies to a lot of disinfectants. The more active they are, the more corrosive they usually are, the more unstable they usually are, so always look up those parameters when you work with them. Also, don't forget, kind of on a side note, to look up the safety sheets, safety data sheets for all these chemicals. They can be extremely, extremely dangerous. The next class, the peroxygens, and a familiar, I'm sure, reagent to you, the hydrogen peroxide, H2O2, commonly employed in households, obviously. They're pretty good disinfectants. Same mechanism, very similar mechanism of action, as I said earlier. So this is like a recurrent theme to remember. A lot of these disinfectants are potent because of the formation of the radicals, the chemical entities that have unpaired electron. So the same with peroxygens and the hydrogen peroxide as the first representative of this class, right? The simplest of them. So they're actually very active. They're very efficient. Decomposition products. So the products of decomposition of these chemicals are not toxic, which is good, and they're biodegradable, which is also good, especially modern day context, right? Uh, Specifically, as you probably know, right, hydrogen peroxide uh, decomposes into water and oxygen. So the best chemicals you can think of, right? Water and oxygen. Um, as far as them being used for the contact lenses, it's okay. You don't have to memorize this. Actually, it's just specifics we don't need. 3 to 6% is usually used for general disinfection purposes. But this 3 to 6% is only good for vegetative forms. If you want these chemicals to be, especially hydrogen peroxide, if you want it to be effective against the spores, you have to elevate the concentration up to 30%. In some cases, elevated temperature helps, increased contact times, all of those factors, don't forget about them. We discussed them 2 weeks ago. Um, Interaction between any disinfectant and the surface is not a magical process from um, cartoonish Lysol commercials where you pour disinfectant and all the bad guys explode. This is not how it works, right? It's a chemical reaction. The chemical reaction does depend, the efficiency of the chemical reaction does depend on the concentration. It does depend on the temperature and the contact time. So those factors are always, you must always keep them in mind. Um, Peracetic acid, this guy here, again, don't need to memorize the formula. This is just to illustrate what it looks like. And one of the representatives of the class of the peroxygens, peracetic acid, is extremely potent disinfectant. Its activity is already high enough, even at such low concentration as 0.3%. It's even sporicidal at this concentration. So as you can tell, a very, very potent chemical, much more potent than hydrogen peroxide. Obviously, only employed for like industrial applications. Another huge benefit, one of the aspects of disinfectants in general, their resistance to bacterial enzymes. Because just like with antibiotics, as we will start studying next week, bacteria when they're being exposed to all these chemicals, not all of the bacteria, but some anyway, are capable of producing enzymes that can decompose the disinfectants, just like bacteria is capable of making enzymes that decompose antibiotics, right? So hydrogen peroxide, being pretty good disinfectant, as you can see, it's actually highly susceptible to peroxidases. So these guys here, peroxidases, these are enzymes secreted by bacteria that can decompose peroxides, hence the name peroxidase. It's whenever you probably are familiar with this concept by now, whenever you have ase at the end, that indicates that this is an enzyme that can kill the respective compound. They can deactivate the respective compound. So in this case, peroxidase deactivates peroxides. So it's good for bacteria, but it's obviously not good for us. We don't want this to happen. So hydrogen peroxide is very susceptible to peroxidases. Bacteria can mutate those enzymes, can make them more efficient. We will discuss those mechanisms in the upcoming couple of weeks in details. But their peroxygens and peracetic acid specifically is very resistant to peroxidases, which is good, which makes it even better disinfectant. So it's a much more reliable disinfectant. It is also not affected by organic dirt as opposed to some other ones. So again, cleaning in this particular case is not as much of a factor because you can just throw it and it will penetrate through organic dirt and still kill the bacteria. Corrosive to metal, pretty potent irritant to us, right? To skin and mucus surfaces in general. So you want to be extremely, extremely careful whenever utilizing these chemicals. So whenever you work with this kind of a nasty chemistry, so to say, always refer to MSDS sheets. Don't neglect it. A very common also misconception. Chemistry especially the one that's being used for industrial applications, is no joke. It's toxic. It can be lethal. It can be whatever. So always know the chemical you work with. Chemicals can be toxic on touch. They can be toxic when inhaled. Some of the worst case, mm, you know, chemicals, right? So always know the chemical you work with, especially if you work with it. Not even I don't even say on a regular basis, just in general, even if it's a one-time use before you touch anything, always refer to MSDS. Ask, people around you working in the lab or something in the facility. Don't neglect the safety. It's not a good aspect to ignore, obviously. The next class, QUECS. Quaternary, know what it stands for, the acronym. Quaternary ammonium compounds. And you worked with these chemicals in the lab. The citrus disinfectant is it's actually the citrus is the one that we use for the benches. When you come into the lab and you spray the bench with the bottle, you have citrus in there, and citrus is a quaternary ammonium compound. So what are those? Again, I'm not asking you to memorize the formula, but for this particular case, at least remember the general idea behind this formula because it will help you understand the mechanism of action much better. It will help you visualize the mechanism of action better. So, as the name suggests, you have substituted ammonium compound. So, this is your ammonia ion instead of hydrogen. So, you have nitrogen in the center. And instead of hydrogens here, instead of being NH4, you have those side chains. The R groups stand for the side chains. You have 4 side chains. Um, that's why they're called quaternary ammonium compounds. Um, what do you need to remember here? Nitrogen in the center is positively charged. Okay. Um, Very important to memorize that. Um, Side chains can be of a different kind. They can be of a different kind of a different chemical nature. Not all the side chains are equally efficient at disinfection. In order for the quaternary ammonium compounds to be efficient as disinfectants, the side chains must must be made of, I guess, chains of carbon that are at least 8 carbons long. Okay, so this side group must have at each of them, all 4 of them, not just one, but all 4 of them must have uh, long C chains. C stands for the carbon here. Must have long C chains of at least 8 carbons long. So this is essential for the antimicrobial effect of these compounds. So if your side chains for your quaternary ammonium compound is like 6 or 5 or whatever, 7 or less uh, carbons long, that kind of a chemical will have very limited bactericidal active antimicrobial effect. Why is that the case? What is the mechanism of action? It's not complicated. Do know it for the test. You need to memorize a couple of things. The mechanism of action. Uh, the quack uses the positive charge of the nitrogen in the middle to attach to the cell wall or the cell membrane of the bacteria. Once it attaches, based on this positive charge, long C chains making up these side chains are responsible for disrupting the cell membrane. That's why they have to be at least 8 carbons long, because if they're shorter, they will not interact with the cell membrane efficiently. So they have to be at least 8 carbons long. So that's pretty much it. That's as much as you need to know. So nitrogen, positively charged nitrogen in the middle is essential for attaching of the whole molecule, whole quack molecule to the cell membrane. And then after that, after the attachment has been a success, the side chains of at least 8 carbons long will disrupt the cell membrane. Without attachment, the carbon chains of the side groups can do anything. Okay, so whenever you use the quacks, they are usually they usually have the highest activity. Well, not usually, but they always have the highest activity at neutral or slightly basic pH. Um, at low pH, it says here they're deactivated at low pH. It's not so much that, well, yeah, you can say that they're deactivated. At low pH, what happens is at low pH, you have a lot of hydrogen atoms in the solution, and those hydrogen atoms are also positively charged, H plus. And what's going to happen, those H plus ions in your solution will block the docking sites, the attachment sites for the positively charged nitrogen on the cell membrane. So positively charged nitrogen, when it attaches to the cell membrane, it obviously will attach to negatively charged docking sites, attachment sites, because positive will require obviously negative sign for attachment, right? So this one is positive, site on the cell membrane is negatively charged. So if you have low pH, your H plus ions from the low pH will shield off, right? They will also attach to negatively charged uh, sites on the cell membrane, and they will therefore shield off that site from the positively charged nitrogen on quark molecule. And because of that, the quark molecule will be unable to attach to this cell surface. If it cannot attach, if it cannot attach to the cell surface, obviously no interaction for the side chains is available, and therefore nothing is going to happen. The efficiency is going to drop significantly. Okay. Same deal with all sorts of anions, organic matter, hard water. They all reduce the activity. Why do anions reduce the activity? They're negatively charged, right? They're not positively charged because smaller anions can also shield off positive charge on the nitrogen atom itself. They can arrange, form this kind of you probably seen it, learned it in chemistry. They form like a cloud around your positively charged nitrogen anions. And then they shield off this charge from the, again, attachment sites on the cell membrane. So it's either the attachment site that can be shielded off or the nitrogen atom itself that can be shielded off. So the best conditions are neutral conditions when there's no additional charged particles, charged chemical entities present in the media, in the surroundings, because either of them can actually shield off the charges, and charges here are essential for interaction. So that's why neutral, slightly basic pH is fine. Neutral pH and no additional ions, no additional molecules or charged particles, rather charged ions in the media is the best, are the best conditions for the quacks. There one more, I guess, downside for these chemicals. They're mostly active against gram positives, not so much against the gram negatives. Why is that the case? And it's a very common case. You must have noticed this trend by now. Well, we discussed this, so we come across this trend very often in the course. Gram negatives in general are much more, well, I don't want to say much more, but on average, they are more resistant to a lot of chemicals. They're more resistant than gram positives. They're more resistant to disinfectants. They're more resistant to antibiotics as well. The reason for it is always this. Again, there are multiple reasons for it, but for now, the main reason you should remember is the double layered cell membrane. Gram negatives have double layered cell membrane, and that cell membrane is very, very hard to breach for any chemical, including the quacks. So that's why gram negatives are so nasty. This is not to say that the gram positives cannot be very resistant species as well. They can be, but on average, again, it's all about stats, so to say, right? On average, gram negatives are more resistant. Quacks are not sporicidal. They're not that efficient. It's nothing like peroxides or aldehyde, simpler aldehydes in general. They're not sporicidal, and they're not active against non-enveloped viruses, the more resistant viruses. So they have limited activity. They're not necessarily the best disinfectants, but they're not as toxic, obviously. The next class we're going to look at, these are not so much the disinfectants, but they're more of a preservative. But I wanted to talk about these in this context anyway. So benzoic acid, sorbic acid, and the parabens. These three classes of chemicals. Again, as it says here, they're widely used as preservatives because their efficiency against the microorganisms is not high enough for them to be disinfectants. Um, but nevertheless, there's a couple important here trends that you need to know. Traditionally, benzoic acid and sorbic acid were employed as preservatives widely before parabens came into picture. However, benzoic acid and sorbic acid are not very good preservatives because they can be utilized by some microorganisms as carbon source. So in other words, they can be utilized as food. Okay, so obviously you don't want the preservatives that can be chopped up by the bacteria for food. And that's when people modified benzoic acid and they came up with the paraben molecules. As you can see, paraben is a derivative of a benzoic acid. It has an additional hydroxyl group in the cycle here, but also it has the side group here. This side group R stands for the side group. So parabens have uh, a lot of good, I guess, antimicrobial properties to them. And that's why they were employed so commonly back in the day. Hopefully not as much these days. You probably heard about it. Um, Parabens are considered to be carcinogenic. Nobody knows to what extent, but they are. But why they were using parabens and what kind of benefits did they provide? A couple of them. First of all, they're not being employed for food as easily as carbon source as easily as the previous two classes, so that's good for us. Second of all, they're not as pH dependent in their activity, so that is especially important for preservatives uh, because of the nature of the preservatives. They are the chemicals that are being added to food products, cosmetic products, all sorts of products, and usually they are expected to act for quite some time, right? Like cosmetic product can last you for quite a bit. Food product, maybe not as long, but nevertheless. Another thing is whenever you use a chemical as a preservative, it is exposed quite often to the environment that is changing its texture, changing its composition, changing its pH much more commonly. So the benzoic acid and the sorbic acid were found to be quite a bit pH dependent in their activity, while parabens are not. So even if the product changes the texture over time, uh, changes the pH over time, parabens are still efficient. So that is very good. So antimicrobial activity of parabens is significantly less pH dependent than that of the previous two. And another third, I guess, good property of the parabens is that the side chain here on top, the identity of the side chain um, governs, I guess, impacts the solubility of these chemicals. And that is especially important for the preservatives. Why? Because especially for cosmetic products. Um, because what can happen, and this is the concept I want you to understand, it's called preferential partitioning. Preferential partitioning. So what can happen in cosmetic products? Uh, they're usually made of several, or well, they're made of different chemicals, but those chemicals can be subdivided into two major categories: oil-soluble chemicals and water-soluble chemicals. And for cosmetic products, it's common that as you use them, they can split. The product can split into two phases. One will be water soluble, the other will be oil soluble phase. And as that happens, uh, as that separation happens of the product. The preservatives, the chemicals that are dissolved in the product, they will follow their favorite phase. So if you have a chemical that is oil soluble, it will follow the oil phase upon separation. If you have a chemical that is water soluble, it will go into the water phase upon the separation of the phases of the product. So why is this bad? Why is this a problem? Because if you have all your preservative uh, separating into oil phase, that means your water phase is completely now devoid, completely free of preservatives. And that means that phase is not protected anymore. And that means that that phase can grow bacteria and that can spoil the product. Okay. It's actually used to be a big problem. So parabens, because of the presence of this side group, they offer this property that if you, very useful property, that if you change the number of carbon atoms in this side chain, you can change their water slash oil solubility. You should know that from basic chemistry. The fewer carbon atoms you have, the more water soluble it is, the chemical is, and the less oil soluble it is. The more carbon atoms you have, the less water soluble the chemical is, the more oil soluble it is. Okay. So with parabens, what you can do into the same product, you can supply parabens, two types of parabens with few carbon atoms in the side chain and a lot of carbon atoms in the side chain. And that one chemical will be water soluble, the other chemical will be oil soluble accordingly, right? So if cosmetic product happens to separate into two phases, oil and water, you will have both types of chemicals to follow both into oil phase as well as into water phase. So in this case, you will keep the product protected, both phases protected. So that is a very, very useful property. And as it says here, methyl, ethyl side groups. So one carbon atom, two carbon atoms are water soluble, 3 plus carbon atoms, so 3 and more, will give you oil solubility. What's the benefit? We just discussed it. So you can provide a preservative system uh, that will protect both oil phase and water phase in case of a separation. And this whole concept of a chemical following the phase, its preferred phase upon separation, that's what's called preferential partitioning. Uh, the chemical separates, partitions, so to say, into its preferred phase based on its solubility. Water to water, oil to oil. Pretty straightforward, right? So that's the benefit of systems like this, preservative systems like this. You can supply two different types of chemicals. One will be water soluble, one will be oil soluble. And then if worse comes to worse, you can have both phases still protected. So that significantly increases the shelf life of the product, obviously translates into better profits and so on and so forth, right? Less expensive products, more profits and so on. But like I said, parabens nowadays are considered to be carcinogenic. Um, so they're being phased out, hopefully. There's a lot of discussion about it, I guess, but you probably heard about it. So the parabens are being phased out. The last, I think, yeah, it is the last. The last class that we're going to look at are the biguanides. The biguanides. Um, these are pretty weak disinfectants. They can be used as disinfectants, but pretty weak disinfectants, and mostly they're used as antiseptics and preservatives. This is the formula. So biguanide group itself is like this. This is the formula of chlorhexidine. That is a very common antiseptic, specific representative of a class. And chlorhexidine is, uh, you probably don't know, but it is a very common, it's the main probably compound in mouthwashes. That's the chemical they use in mouthwashes. Its mode of action is very similar to that of Quex. It has nitrogen, and this nitrogen can gain form a dication, as it says here. It can form a dication. It can form have positive charge, and that positive charge will be looking to attach to negatively charged sites on the surface of the cell wall and the cell membranes of the bacteria. Okay. In order for this to happen, same deal. In order for this to happen, the attachment has to occur. The successful attachment has to occur. So these chemicals are sensitive to pH. Okay. Sensitive to pH, you want to be using them in the solutions of neutral pH, pH of 7, around 7, maybe slightly basic, and without presence of any other anions, cations in general. So no hard water, no soap, nothing, no organic matter. Activity can significantly drop because of this mode of action. It's not sporicidal, like I said. It's not necessarily very effective, so it's mostly preservative and antiseptic class. Not effective against problematic bacteria. Again, don't forget what those are. Those are problematic species that show elevated levels of resistance. So Mycobacterium tuberculosis, some Pseudomonas species, E. coli can fall into this, definitely falls into this category. There's a lot of nasty E. coli species. Staph aureus can be that as well. The famous MRSA strain from the past. Methicillin-resistant Staph aureus. Don't have to memorize it for now, but we will cover that in the next lecture. So they're not very effective against those, so hence only antiseptic use and preservative use. Can be used in ophthalmic products, but whatever. We're not going to go into the specifics. You don't have to memorize that. Remember the mode of action. Remember the fact that they're not necessarily the strongest one. So they are generally considered to be bacteriostatic agents. They can be cidal as well, so they can reduce the concentration of bacteria, not just sustain it, but reduce it as well. But there are better chemicals for that. Okay. And that's pretty much it. So these are the classes we're going to cover, specific ones. Like I said, all of these last slides, I don't know, quite a few details, not that many. I obviously put only the most essential data. There's a lot more that can be said about each class. There's no way we can cover all the classes. There are dozens of them. You don't need it. I was just trying to show you the most common classes, some of the most common mechanisms of action. Make sure you understand the mechanism of action. If you understand the mechanism of action, you can always navigate in science much better. If you just memorize things, you're never going to have enough memory to memorize everything. Okay, you're not a machine, nor does the machine have infinite memory, right? So you must understand the mechanisms. If you know that, it's much. You're much better off. So there will be questions about the mechanisms. The mechanisms are pretty basic. Two mechanisms, basically oxidation and then of the cell wall and the membrane, and then attachment with subsequent disruption of the cell membrane and the cell wall by these side chains, long carbon chains of 8+ carbon atoms, pretty much. And everything else, like the nasty disinfectants like peroxidants, they're just oxidizers. They're just oxidizers. They form free radicals. Those very reactive chemical entities with unpaired electrons. Okay. Alrighty, guys. So that's gonna be it for today. Next week we're gonna start antibiotics. Pretty extensive lecture. There will be a lot of information there. I strongly suggest you attend the last two lectures. There will be a lot of information. Not a lot. Again, don't want to scare anybody off, but there's going to be quite a bit of information, especially if you never kind of studied these kind of topics before. We will talk about the antibiotics, a bunch of classes, some general considerations, as well as specific classes, mode of action, how they work, what they do, and so on and so forth. And then after that, we will cover the antibiotic resistance, how bacteria responds to that, and how pharmaceutical industry addresses those issues. We will talk about some problems associated with pharmaceutical industry and their stance on antibiotics. We'll discuss quite a few actually interesting concepts, not just science related, but also general why we have issues with infectious diseases these days, why the infectious diseases are making a comeback presently. To an extent, obviously, it's not as bad as it used to be, but 200 years ago or something. But nevertheless, it's on the rise, right? All these pandemics and whatnot, it's kind of prove that, right? So that's next week. All right. So alrighty. That's it for tonight. Have a good weekend, and I'll talk to you next week. Send me an email again, as I said earlier, if you want to see your test. Next week I should be in college. We can schedule this. Uh, don't neglect the chance to take a look at your mistakes, especially if you didn't do so well on the test. Alrighty, guys. Have a good weekend. Talk to you next week.
Thank you, Professor.